just like our tube of cells. Okay, so we're gonna walk over here again, and just to be cautious, we look at it as the same amount. Now we have to put it, this is at 16, and we have to put it directly across so we can see it's balanced, and just make sure it is by doing that. They're directly across from each other. Is there a lid that goes on here, Ron?
No.
So we're going to close this. And the fact that this is a large centrifuge for our cells, we need to centrifuge them at a lower speed, because a higher speed will lyse the cells and kill them. So what we want to do now is we want to start a centrifuge. I've never used this before, Ron, so can you assist me with it? Oh, it's just start.
Yeah.
So we're starting it, and we're waiting for it to get up to speed. It's actually going to spin for 5 minutes. We're trying to see if this is an appropriate speed to actually get the cells to come down. If it is, we'll use this from now on for this process, and then we'll proceed to actually plate the cells. So what we're going to do is we have 5 minutes. In that 5 minutes, we're going to prepare the plates to actually plate the cells on new plates. So we'll do that now. Now, Ron, how do you want to plate these cells? Do you want to plate them on? So I brought some plates for you. We can just do 2 of these.
Yeah.
All right.
This is probably better.
So what we're going to do, there's multiple places that you can do this. This is actually a plate that actually reduces contamination with the way it's made. This plate is used for cells that don't have to attach to the bottom, of free-floating cells or cells that attach. So this is just a different way to put the cells in a different container. However, we're going to switch to this petri dish type of tissue culture dish, and we're going to label them and get them ready to put cells in. Alright, so we're going to walk over here, and one of the biggest things in science is labeling things. So one thing I taught Aberroni visit or came to our lab over the summer is to always label everything that he does. Write it down. So these cells are, the cell line is RAT2. We need to know what the cells are. And Aberon, what's your initials?
A.
AK? Yeah, that's cool initials. Alright, so then we have the type of cells, who actually is taking care of the cells, the date, which is March 11, 2014.
It's the 12th.
Yeah, it's March 12th. Thank you, Aberon, for correcting me again. I thought I was 11. Alright, so then we're going to label both of these plates with the same things. And we know exactly what the date is. Now, if we need to add like a reagent or anything like that, we will actually put it on the plate too, so we'll know exactly what we have. So we have 2 tissue culture plates. I said before that these plates hold a maximum of 10 mils. So we're going to put the 10 mils there. And the good thing about science is that we have a lot of incubation periods. When you have incubation or centrifuge periods, that's when you go and set up the rest of the things you need to do. That way you can work very efficient and effectively when you're doing your science, because science is all about time, so you have to make sure that you're good at managing your time when you carry out these experiments. Alright, so now I'm going to add the 10 mils of medium to the plate. Put it back on. You see how that medium is nice, and it's a nice color. That's the color that we want. Like I said before, if that color gets to be more of an orangish, yellow-orange color, that means that that's an acidic environment, and either the cells are overgrown, it can be overgrown cells, contamination, or the media just needs to be changed because the cells have used all the nutrients in that medium. Alright, so I'm just putting the cells, the media in the plate for the cells. Alright, so we have that. Take that, put the top back on, and we have our 2 plates that we're actually going to put those cells in. Alright, so once again, we've done that. We don't need the trypsin anymore, so anything that I don't use in the hood, I remove from the hood. We remove that, and now we have our nice, neat environment to actually do the rest of our experiment. Sometimes I'll just wipe, just make sure everything's nice and neat. Put that there. Any trash, we do the same thing. Get it out the way. And now we're waiting for the centrifuge to be complete, and then we're gonna put the cells on. So we're gonna use this pipette. We'll always do, like I said, we need to make sure that that pipette is sterile because we're going into the medium. So put enough on there. As you can see, I use a lot of ethanol, and the reason that we have to use more than we want to use is because this hood here is it's a makeshift tissue culture hood, but it still can be very effective if you or the person that's doing the experiment practice sterile technique. Sterile technique is very important. So anytime I do something like that, this is the part we want to use. So I always wipe it off, and I'll put it like this. That way I don't disrupt that part. So once again, I always get everything set up. These tips are autoclaved, so they've been sterilized, and I can use these tips in the tissue culture hood. Autoclave means that it's a heated process that kills all the bacteria or whatever contaminants might be in these tissue. In these tips. So what we always do, don't take the whole thing off. You know, put some on there to let people know, hey, this is sterile. That's been autoclaved. All right, so now we've done that. We have everything kind of set up for us. What I want to do now is go check and see if the cells are done. So we're going to go back over to the centrifuge and see what we have.